everyone. Let's go into what we have today.
Every second, millions of DNA and RNA molecules are being synthesized and broken down inside your body. But what happens when these pathways go wrong? Welcome to the fascinating world of nucleic acid metabolism.
Every second, billions of cells in your body are building DNA, repairing genetic damage, and producing RNA needed for protein synthesis. But where do these nucleotides come from? How are they recycled? And what happens when this pathway fail? In today's tutorial, we simplify nucleic acid metabolism from synthesis to degradation and uncover the diseases linked to this pathway. You will see the cinematic view into the human body, traveling through blood vessel into the cell nucleus. Nucleic acid are the molecule responsible for storing and transmitting genetic information. I don't know whether you know that. DNA stores hereditary information, while RNA plays a vital role in protein synthesis. Both DNA and RNA are made up of repeating units called nucleotide. And you ask, what's a nucleotide? A nucleotide consists of three components: a nitrogenous base, a pentose sugar, and one or more phosphate group, as you can see in this image here. Image is comparing the DNA and RNA structure. And now in purine and pyrimidines. The nitrogenous bases are classified into purines and pyrimidines. Purines have two fused rings and include adenine and guanine, while the pyrimidines possesses a single ring and include cytosine, thymine, and uracil. Understanding this structure will help you very well in metabolism understanding. In other words, understanding this structure is essential because their metabolism differs significantly. De novo purine synthesis. The novo purine synthesis begins with ribose 5-phosphate derived from the pentose phosphate pathway. The ribose 5-phosphate is converted to PRPP or phosphoribosyl pyrophosphate. The committed step is catalyzed glutamine PRPP amidotransferase. After several energy-consuming reactions, inosine monophosphate or IMP is formed. The IMP serves as a precursor for both AMP and GMP. Now we are quickly entered into purine salvage pathway. Instead of synthesizing purine from scratch, the body often recycles them through the salvage pathway, and this pathway conserves energy and efficiently reuses purine synthesis. The enzyme HGPRT salvages hypoxanthine and guanine. Deficiency of HGPRT leads to the development of Lensnan syndrome, characterized by neurological abnormalities and self-injurious body, like you can see. In purine degradation and uric formation. When purine are no longer needed, they undergo degradation. Adenine and guanine are converted into xanthine. The enzyme xanthine oxidase then converts xanthine into uric acid. And you will notice that excess accumulation of uric acid can result in crystal deposition within joints, causes gout, as you can see in this image. This is a biochemical animation of purine breakdown. What you can see here is the nevo-pyrimidine synthesis. Unlike purine, the pyrimidine ring are synthesized first and later attached to ribose phosphate. The process begins with carbonyl phosphate synthesis too. Eventually, uridyl monophosphate or UMP is formed. And what this UMP does is that UMP gives rise to other pyrimidine nucleotide, including the CTP and thiamidine derivative. You can see the scientific pathway diagram of pyrimidine synthesis showing carbonyl phosphate formation through UMP production. After that, you look at pyrimidine degradation. When you look at pyrimidine degradation differs significantly from purine degradation. The end products are highly water-soluble compounds such as beta-alanine and beta-aminoisobutyrate. Therefore, pyrimidine breakdown rarely leads to crystal-associated diseases. When you look at this image, the educational infographic illustrating pyrimidine degradation products entering the metabolic pathways. One thing I want you to know and understand is that there is clinical disorder of nucleic acid metabolism. Disorder affecting nucleotide metabolism can have devastating effects. Examples include gout caused by uric acid accumulation. We have orotic aciduria, which results from defects in pyridine synthesis. Orotic aciduria results from defects in pyrimidine synthesis, like I mentioned. And another important condition involves adenosine deaminase. This deficiency in adenosine deaminase contributes to severe combined immunodeficiency, and understanding this disorder bridges biochemistry and clinical medicine. Then there are medication that work by altering nucleotide metabolism. If you go to allopurinol, which inhibits xanthine oxidase and reduce uric acid production, then we have methotrexate also, which interferes with folate metabolism and nucleotide synthesis. This again is the picture, as you can see, nucleic acid metabolism master concept map. You can see it here for you to zoom it and see what it's talking about, because it will help you in better understanding of how these pathways are intercalated in different ways. So let's summarize this. Purines can be synthesized de novo or salvage, while purine degradation can end with uric acid formation. Pyrimidine follow a different synthetic route and degrade into soluble products. Defects in this pathway lead to important clinical disorders. Several medications exploit these pathways to treat diseases. So you can see the master concept, like I said before, summarizing the nucleic acid metabolism. And these master concepts include the purine synthesis, salvage pathway, degradation pathway, pyrimidine, and others. So in conclusion, nucleic acid metabolism is far more than a collection of pathways to memorize. It explains how our bodies build DNA and RNA, recycle valuable resources and eliminate waste products, and also respond to life-saving medications.
Nucleic acid metabolism isn't just about memorizing pathways. It's the foundation of genetics, medicine, pharmacology, and disease. Master these concepts and you'll unlock one of biochemistry's most important topics. If this tutorial helps simplify nucleic acid metabolism, don't forget to like, subscribe, and share this video with your classmates. What biochemistry topic should we tackle next? Let me know in the comments.